Hi, this is Mrs. Bustamante. Today we're going to talk about karyotypes. We're going to talk about what they are, why are they used, what do you need them for, how do we know if a person has a chromosomal disorder, and how do we know if that person is male or female based on the karyotype, what genes they have, if you will. So a karyotype is just a picture of a person or a fetus's chromosomes taken when they're in mitosis. We have to take them in mitosis because that's when we see them as condensed chromosomes and they're nice and easy to compare, or easier, I should say. It can tell us if there's a chromosomal disorder, meaning if a chromosome is missing sometimes even a piece or an entire chromosome, or even if there's an extra chromosome. And it can tell us the sex or based on the genes that are present, right? Are there two X's that make it a female? Are there two X chromosomes? Or is there an X chromosome and a Y chromosome, which is what differentiates between that male and that female? All females will give one X to their offspring, and remember, males can either give an X or a Y, so we say that there's a 50-50 chance, right, of when they have an offspring, it being male or female. Today we're going to look at karyotypes for humans. And remember that when we look at karyotypes for humans, or how many chromosomes humans have, they have 23 pairs or 46 chromosomes. And in a normal karyotype, we'll see that each of those pairs, each of those 23 is a nice pair. And typically we match up these chromosomes based on the length of them, based on where those centromeres are, and based on what we call the banding pattern of them, or where there's like dark spots and light spots, because that means they probably carry similar genes. Now, we organize those chromosomes when we're looking at a karyotype in a nice order. Typically, it's about biggest to smallest. And our last pair here, or pair 23, is the sex chromosomes. And this is where you can tell if it's male or female. If it's female, you're going to see two X chromosomes here that look about the same. If it's male, you're gonna see an X chromosome and a Y chromosome, and that would tell you that it's male. And yes, typically the Y chromosome is a little bit shorter than the X. They don't look the same, and that's how it's supposed to be. So, right, when you take a karyotype, you're not going to see them all organized like this when you just originally get the chromosomes and start looking at them. They're going to be all mixed up, and you got to match them together as best you can, and then look at similarities and differences. So in a normal one, what do we want? We want two of each chromosome, right? So we have 23 matching pairs in humans, and they should be about the same. And remember that 23rd pair is always the sex chromosome that you should see. So what if it doesn't match up exactly as it should? Because sometimes when you're going through meiosis and mitosis, those chromosomes won't divide as we hoped, right? There's some disjunction, we call it, that happens. And probably the one that we're most familiar with is something called Down syndrome, or trisomy 21. And we just talked about that each pair of chromosomes, you want to have a pair. So there's just two of them. But if you see a trisomy, that means that you have three chromosomes at one of those positions. Down syndrome is called trisomy 21 because it has three chromosomes at the 21 site. It has an extra chromosome at that 21 site. So people with Down syndrome, they do tend to survive and live on to be born, right? Sometimes they have some intellectual disabilities or some hard time processing as quickly as somebody with just two chromosomes at 21 would have. Sometimes they can have some different facial appearances, weak muscle tone. But typically, they always have some sort of intellectual disability, whether it's mild or moderate. But for the most part, they can live pretty happy lives, but we know that it's a bit different. So there's where you have an extra chromosome at that 21 position. Karyotype syndrome is also known as 5p minus syndrome because it's actually missing a part of this fifth chromosome, not the whole thing, just a little bit of it. And infants with this condition often have a high-pitched cry that sounds kind of like a cat. I'm guessing that's where the name comes from, because this is cat in French, right? This disorder is characterized by intellectual disability, delayed development, sometimes microcephaly, where it has a smaller head size, lower birth weight, weak muscle tone, um, and they can also see some wider set eyes, lower set ears, smaller jaw, rounder face. And sometimes the children with this can also be born with a heart defect. We can see a heart defect in them. So again, this is missing part of the chromosome at chromosome set 5, not the whole thing, right? Which is still even problematic. We need all that information at all those chromosomes. Turner syndrome is a chromosomal condition that affects development in females. It's because we're missing an X chromosome here. They only have one X chromosome at that 23rd position. Typically, they're shorter in stature, and this becomes evident somewhere around the age 5. They have an early loss of ovarian function. Is also pretty common. Those ovaries tend to develop normally at first, but egg cells or oocytes usually die prematurely and make it difficult to go through puberty, right? Unless they receive some hormone therapy. And a small percentage of people with Turner syndrome retain normal ovarian function through young adulthood. But not all of them, right? So that's missing an entire chromosome. Happens to be a sex chromosome because it's at that 23 pair. It's missing that extra X chromosome. Then we have Patau's syndrome, which is caused by a chromosomal abnormality at set 13. And we could say, again, this is a trisomy, right? Because there's 3 chromosomes at set 13. The genetic material disrupts normal development, causing multiple and complex organ defects typically. So it can occur either because a cell contains a full extra copy of chromosome 13, or because a cell contains an extra partial copy of chromosome 13. Caused by a incorrect disjunction during meiosis, right? Or non-disjunction during mitosis, basically when we're splitting apart the cells. And then we have Edwards syndrome. Edwards syndrome is known as trisomy 18, and it's a genetic disorder caused by the presence of all or part of a third copy of chromosome 18. And many parts of the body are often affected by this. Babies are typically born small and have heart defects, and other features might include a small head, a small jaw, clenched fists, overlapping fingers, and sometimes even severe intellectual disability. So we can really see the importance of having just the right number of chromosomes, right? You don't want to have too many or too few. And sometimes when chromosomal disorders are so big or important, I guess you could say, or massive might be a better term than important, that you would see a fetus maybe not even live to full term because they don't have enough information to survive. So really seeing the correct chromosomes in the correct numbers is really important for development.